Uh, Hello everyone. We are going to commence this month webinar conducted by Institute of Physics Sri Lanka. I am Shanta Gamuge, President of IPSL. The main objective of the IPSL is to promote and advance the science of physics in Sri Lanka. Before introducing our resource person today, I would like to warmly welcome all the participants of this webinar. Thank you for making the time to watch our seminar today also. So today we have invited Dr. Pubuditha Abeyasinghe, a postdoctoral research fellow in computational neuroscience at Monash University, Australia. Let me briefly introduce herself. Dr. Pubuditha obtained her BSc special degree from University of Sri Jayawardenepura with first class honors, and MSc and PhD from University of Western Ontario, Canada. Expertise include computational modeling, data analysis and interpretation, and the development and implementation of research tools. So today she will talk on modeling disease progression of Huntington's diseases. And so without wasting time, I would like to invite Dr. Pubuditha Abeyasinghe to give your presentation. Thank you. Over to you, Pubuditha.
Thank you for the introduction, Professor Shanta. Let me share the screen. Can you see the screen?
Yeah, yes.
Okay. Thank you very much for the introduction, sir. And also thank you very much for inviting me to present my work for IPSL. As Professor Shanta mentioned, I'm a computational neuroscientist working in at Monash University, and I'm studying about the Huntington's disease. So the objective of today's presentation is to give you a brief introduction and a brief idea about the work that I do at Monash. Okay, so this is the flow of my presentation. For the first half, I'm planning to tell you a little bit about the disease, Huntington's disease, what is it, and the disease progression, and also why I'm studying about this. And for the second half, I will be introducing some of the work that I do very briefly. So what is the Huntington's disease? It is a genetic neurodegenerative condition. And everyone, you and me, has 2 copies of Huntington gene in our DNA, that contains a section of repeated genetic letters known as CAG repeats. So as you can see at the top here, these are the CAG repeats. So in every person's Huntington gene, we have these CAG repeats. Now, Huntington's disease, in short HD, occurs when one copy of this Huntington gene has more CAG repeats than a certain threshold. And that is the case that's shown at the bottom. So the number of CAG repeats here are more than the double compared to a healthy person. Now, what is the threshold of this number of CAG repeats in order to get the Huntington's disease, or in order to be diagnosed with the Huntington's disease? If we have 26 or fewer number of CAG repeats, then that person will not get HD. However, if the number of CAG repeats are more than 36 or between 36 and 39, there's a possibility of getting HD. And if the number of CAG repeats are higher than 40, that person will definitely get HD at some point of his or her life. These are the most common symptoms of Huntington's disease. It's characterized by progressive decline in movement. So a person with HD will lose the control of their movements eventually, and thinking ability will change, and they will have personality changes, behavioral changes as the disease progresses. And the disease strikes mostly between 30 to 50 years of age. That's one of the tricky parts of this disease, because a person will be born with the gene mutation, but they won't get the disease until later their lives. So our objective is to try to see whether we can bring the diagnosis window a little forward, so that we can act fast to modify the disease progression. Now, there are three basic stages of disease progression. If you look at the focus on this blue color stripe, the first stage is pre-manifest, then you have the early manifestation, finally the manifestation of the disease. This is the basic stages of the disease. One key point is, in HD, the pre-manifest stage is quite long. Because of that, it results in diversified biophysical and physiological factors across the spectrum of this disease, which again results in a widely varied symptom profile. I will explain what I mean by widely varied symptom profiles in a little bit. But for now, let's focus on the groups of these disease, stages of this disease. So, as I said, these three stages are the stages that a patient will be diagnosed on. So, if there's a person who is getting some symptoms and they go into a clinic to get tested to be diagnosed with the disease, and the physician or the person who's trained to diagnose the disease will give that patient some tests, and they will, depending on the results, they will decide, okay, this person is at the later stage of pre-manifest stage. But the problem with this categorization is it's subjective. So if I do the diagnosis, I would say that this person is at the later stage. But if you give them the same tests, identical tests, you will evaluate the test in a slightly different way, because the way we think are different. So you might diagnose as that person is at an early manifest stage. Because of this ununiformity, we need to find a better way to categorize these patients for our research purposes. Therefore, the most commonly used categorization is called as CAP groups. That's what I have at the top. So CAP groups are categorized using this equation. So we can calculate the CAP score defined as this, according to this equation, which depends only on the age of the person and the number of CAG repeats. So it's not going to be subjective. If I do the calculation, I'll get the same CAP score as you do the calculation. So doing this calculation, we get a CAP score. Then using these commonly known threshold values, we can categorize the patients into three different groups. Here I have four groups, because CAP group 1 is always reserved for controls, because always we do things in comparison to a control group. With these CAP groups, CAP 4 is for the people who have highest progression of the disease. Now, there has to be a way to diagnose these people into these groups. So that's when the markers, biomarkers, come in. On the left side, I have the primary biomarkers, which means if when a patient goes to a physician or a trained personnel to get tested or to get diagnosed, they will give different tests that are labeled here. So we have single digit modality test, Stroop test. Those are cognitive markers, which means they measure your thinking ability to do things like solve problems, mathematical problems, and so on. These tests, total functional capacity, total motor score, these two tests measure called as clinical markers. Again, they will measure the ability, the motor ability of doing motor tasks and so forth. Then we have psychiatric markers. So these are the primary markers that a physician or a trained personnel will use to diagnose a person, whether to decide whether they have HD or not, and if they have HD, at what stage are they at now. Simultaneously, we have the volumetric markers or imaging markers, we say. So while at the same time, so we do these tests, and then they take a MRI image of the brain, and we can measure the volumes of separate regions of the brain from that MRI image. And these are also used as markers, but they are called as secondary markers. So in my research, we are using all of these markers to do what I do. Now, what do I do? Our objective is to find a better stratification method to identify different symptom profiles. I will explain what do I mean by different symptom profiles. But before that, the stratification methods. As I said in the previous slide, we have already two methods. We can categorize the sub-HD individuals into pre-manifest, early manifest, and manifest. That's one way. Or we can use the CAP groups. That's another way. But in both of these ways, it does not reflect the true symptom profiles of all the subjects. It's a general idea of a group of subjects. But that's not enough for our purpose. So I will try to explain what do I mean by symptom profiles using this graph. Here I have, let's say we have three subjects in these different kinds of lines. I am representing three subjects, and three colors represents three different markers or measures that we use to measure their cognitive clinical ability. On the y-axis, I don't have a scale because this is just for the illustration purposes, and I can't put all these three markers on the same scale. So if we look at the shaded area, and let's take SDMT, single digit modality test scores, for an example. For subject, we can see that the score is declining or decreasing continuously. And second subject also has a decreasing trend, but the rate of decrease is different in these two cases. When it comes to subject number 3, we can see that until up to 4 years, there is no change in the SDMT score, and then only it starts to decline. Similarly, if we can look at the total functional capacity, a clinical measure, we can see that the first subject has a continuous decline, and the second subject has a sharp decline, and the third subject again has a continuous decline. If you look at the putamen volume, again, first subject has a sharp decline until up to 3 years, and then it saturates. Subject number 2 has it doesn't change until up until 2 years, and then it starts to decline. So what I wanted to show from this graph is that not every subject shows the same kind of symptoms with the same pattern or even a similar pattern. There are some people, some subjects who has HD shows cognitive decline at the very early stages like this, But there are some subjects who doesn't show cognitive decline until later of the time span, but they show volumetric decline at the very beginning. So this is very important when we try to develop treatments for the disease, because if we develop a treatment that will help to reduce the volumetric decrease, and if we give that to a patient who doesn't show volumetric decline but shows a cognitive decline, we might not get the most efficient results from that. And that because of that, it's a very important part of this work to find a method to identify these like subtle differences in symptom profiles. That's why I do what I do. We have, thanks to the principal investigators of these three data sets, we have a huge data set altogether, which comprises of 3 separate data sets. First one is Image HD. It's an 18 months long study. It was collected at Monash University itself. There were 108 participants. Then we have the second largest study called as Track HD. That study was 7 years long, and it was collected from 32 sites from UK, US, Canada, and Australia. And then we have the longest and the largest study, Predict HD study. It's 12 years long. The data was collected from 4 sites, and there were 1,162 participants. So the one objective of my work is to combine all these data sets and study about the combined data set, which represents a big community that are coming from different geological locations, and also they are at different stages of the disease as well. So each of these study cohort have set of healthy subjects as well as a set of HD patients that are in different stages of their disease. And for all these data sets, so these are longitudinal data. What happens is they start the study. All the participants are being diagnosed and tested. They have they give the test to these participants, and all the data is collected, and the MRI images were taken at the first time point. And then most of these studies are annual collections, so they've been asked to come to the center after 1 year. So that's why this is 12 years long, because they have 12 data points for a set of subjects who continuously participated in this study. So for all of these subjects, at every time point, we have all the clinical cognitive psychiatric markers that I explained in the previous slide. And also we have the MRI imaging data. So using this, my objective is to find a computational method or a computational model to better stratify the participants and find different symptom profiles. Now, in order to do that, this is my first project that I conducted using the multivariate linear mixed model. So the idea for this project came from the idea that so we have different markers, as I explained before. Like we have clinical markers, cognitive markers, psychiatric markers, and volumetric markers. So in previous work, most of the time, 99.9% of the time, what people do is they study about each of these markers separately, and we can learn, like we have learned a lot of things about each of these markers separately, how they progress, like what happens with time and all that. But the idea we had was we wanted to see whether we can combine these markers and whether that will give us a better understanding about the disease. In order to do that, we used multivariate linear mixed model. I'll give you a very brief explanation about the model. A linear mixed model is a model that we use to model an outcome variable. So why is an outcome variable that we want to model? For an example, the symbol digit modality test. To model that, we use predictor variables. For an example, the disease stage or the CAP group and time. And then we use fixed effects, coefficients, and we have random effects and all that, and then an error term. Also, we control this modeling, linear mixed model for gender and education. Now, that's the linear model. We have a variable, we want to model that, and we can use the linear mixed model. But in our case, we want to combine different variables and model the progression. That's where the multivariate linear mixed model comes in. So let's say we have three different variables that we want to model. We want to see how these variables change with time. So we can model like each of these is a separate linear mixed model. But in multivariate linear mixed model, what happens is, let's say Y1 is SDMT, symbol digit modality test. I am going to model this using the same strategy as in linear mixed model, but this time I want to control the effect of the other variables like Stru and putamen. So I model one by controlling the effects of other variables. It's a very complex model because it has the time component as well, but it's very effective. So that's what the multivariate linear mixed model does in very brief terms. Using this, I have conducted my first project, and we have published our work as well. To give you an idea, so we have the 6 markers, biomarkers, and we want to, as I said, we want to combine these markers and see whether we can find one model that's sensitive for the disease progression than using single variables. So by combining all these biomarkers, I came up with 63 different models, so 63 different combinations, and I used multivariate linear mixed model. So I simulated MLMM for each of these cases and tried to find the best or the most sensitive. We have already published the results from that in the Journal of Movement Disorders last year. Now, I'll show you one of the results just to keep things simple. This is one result that I'm showing, just one for the combined data set. What I have shown here is DIC is a statistical parameter that we can use to compare different model fittings. So lower the DIC, higher the sensitivity is, or the better the model is. Now, each of these points is one of the 63 models that I explained in the previous slide. So for an example, if I go here we have, this is one model that contains putamen and codeine. And if I go here, this model, this is one model that contains SDMT, symbol digit modality test, and total functional capacity. So by comparing the DIC value of these models, we were able to show that volumetric markers, which are putamen and codeine, are the most sensitive ones for disease to track the disease progression compared to the other clinical markers. This is a novel finding because earlier the idea that we had was that the clinical markers are better than the volumetric markers. However, we showed it otherwise. And one explanation for that is, as I said, the clinical markers are very subjective. Like it doesn't change a lot, it doesn't vary a lot, but it's subjective compared to the volumetric markers, because clinical markers always a physical person will be doing the evaluation, and the patient has to answer as well. So it's subjective compared to the volumetric markers. Because of that, we concluded that volumetric markers are better to identify or track the disease progression in HD. Moving on to the second project. Now, it involves calculating the brain predicted age. So it's a common understanding that human brain shrinks as we age. Also, human brain can shrink as a result of some brain diseases, like in HD. That's why we see volume decline as the disease progresses. Now, the objective of this project is to investigate the feasibility of this brain predicted age to use as a stratification parameter or stratification parameter of the symptom profile, so HD patients. Now, the way I'm doing it is, our collaborators have developed a machine learning technique that takes into account, takes in the MRI image and calculates the age depending on that brain image. So the point of this is, if we take a healthy subject, the chronological age, actual age, won't be significantly different from the brain predicted age. So the age that calculation that comes from this machine learning technique, we call it brain predicted age. So the general idea is, if the person is healthy, the difference between brain predicted age and the chronological age will not be significant. However, when the disease progresses, the brain shrinks. Therefore, the difference between the chronological age and the brain predicted age will be different. So this is the hypothesis that we wanted to test. One of the hypotheses that we wanted to test using this method in this project. In doing so, this is a very initial result. I'm still working on this project. So here, what I'm showing is the brain PAD. Brain PAD is the difference between the chronological age and the brain predicted age. So we were able to show that the brain PAD, the difference between these two ages, are not significantly different for the healthy subjects. That is for CAP group 1. And then we were able to show that this brain PAD increases as the disease severity increases. As we go this way, as the disease severity increases, the brain PAD increases, which is a good sign, because which means we can use the brain PAD or the brain predicted age to track the progression of the disease. I'm still working on the rest of the analysis, so this is just to give you a basic idea of that. That brings me to the end of the explanation of my work. So in order to summarize, HD is a neurodegenerative disease, and there is no treatment for that disease yet. So that's why it's very important that we work on better stratification methods, so that the pharmaceutical companies can conduct trials efficiently and find some treatments or treatments for the disease. So our objective is to find a computational method or computational model to better stratify the symptom profiles of HD patients using the complete data set that we have access to. And the identification of an effective stratification method of HD will be significant for therapeutic development for two reasons. First is, it will allow the selection of participants with the specific symptom profile. So to explain that further, let's say we have a group of HD participants. In that group, we might find that there are some participants who shows a cognitive decline early in the disease, but saturates later. And then let's say that there is another group of participants who doesn't show any cognitive decline, but who shows a volumetric decline earlier in the disease. So if we treat both of these groups as the same, we might not get the outcome we want because their symptom profiles are different. They do not react in the same way to this disease. So it's very important to identify and separate these two groups from the big sample. And secondly, it will aid in finding an optimal window for pharmacological interventions. Again, coming back to the same point, if we let's say we develop a drug that will slow down the progress or slow down the volume decline of the brain. So we have a tablet that slow down the volume decrease in the brain. So if in order to use this drug, we have to know we have to have a group of people, we have to have a sample whose brain decline has already started. So we have to find the specific window that the brain decline or the volumetric decline has already started. Otherwise, we won't get an effective result from that drug either. So it's very important that we separate the people with different symptom profiles, and also we select the optimal window for pharmacological interventions in the future. And that's why that's where the computational modeling part comes in to do all the work. And that brings me to the end of my presentation. I hope you learned something from the last half an hour. So I'd like to thank my research group in Monash and also in the US, and also the principal investigators of all the 3 studies who made all the data available for us to use, and CHDI Foundation, and also the funding sources. Lastly, thank you very much for your attention.
Thank you very much, Dr. Pubuditha. Now it is open for the discussion. So if you have any question, you can ask Dr. Pubuditha.
Pubuditha, is there any resemblance between Huntington's disease and Parkinson's or Alzheimer's?
All of those 3 diseases are neurodegenerative diseases, but they are slightly different in some way. So the Alzheimer's disease, the primary cause is it attacks to the memory, not the motor movements. Whereas the Huntington disease, its primary result is decrease in movement. It also affects the psychiatric ability. Whereas in the Parkinson's disease, it's mostly affecting the movements. So Huntington's disease is kind of a combination of the other 2. As a whole, they are all neurodegenerative diseases, but the difference is the primary the way they react. The disease, the way disease progresses, not react. Yeah.
Thank you.
Dr. Pubuditha, thank you very much for your nice talk. Actually, I wondering to know about now most of the studies now they are utilizing neurofilament light in clinical trial in HD. So how about incorporating the neurofilament light levels and Huntington level in this model that you have done here?
That's a good question. Until now, I haven't incorporated any of that, but there's another student in our group who's initiating that now. So I'm not sure about the results or anything. It's still in the very early stages, the work. but we are doing that.
Thank you.
Thank you very much. And there is another question in the chat box.
Okay.
Could you please explain that? Have you checked the effect of genetic modifiers and epigenetic changes in a model?
Yeah, I haven't checked these things. They are mainly like biological factors. My work is mostly on the clinical, on the data set that has already been collected. So for that data set, we don't have these genetic like information for the genetic modifiers and epigenetics and stuff. So I haven't done that.
Ask a question or comment. I'm Ranil De Silva. Actually, I run the Huntington disease clinic throughout the country, from north to south. And Lakmal is my PhD student who just asked you a question. So basically, we give a free of charge genetic testing for Sri Lanka, all the patients. So we have the biggest biodata, Jane, as Lakmal said, with US NIH funding, we have done the biomarkers. So I just want to know, I mean, if you could have we could have a later on a discussion with Lakmal and Nalika. These are two my PhD students. I mean, because I mean, from a biological aspect, we have the all the motor functions, the psychiatric functions. We have actually seen the progression. We have the genetic testing. We have the proteomics, metabolomics. So therefore, I think if Monash, actually, I have been in close touch with Professor Julia in Monash. So actually, so I mean, if the interested parties could have a later on, Lakmal could organize a Zoom meeting with your doctors in Australia or in Julia. I mean, I have been or Julia because I have visited Monash and given a talk also in Monash with Julia. So therefore, I mean, if you all are interested, we could see how best. I mean, we have the data, and Lakmal is just finishing his PhD. He's a final year student and writing his thesis. So we can just see how best we could work out something there.
Yeah, we could actually. I am currently in Sri Lanka, so I could meet you in person also if we can find a time. We can do that.
That's good. So I mean, how if you can just put your email or something, then Lakmal could contact you.
Yeah, I will do that. I will have my email in the chat box. I will send it to you actually. Yeah.
Mine is ranil.sjp.ac.lk.
Yeah, I just sent the email.
Thank you. Any other questions or comments? So I have one question regarding ML results. Actually, that is a theoretical one. I couldn't understand that the graph you show DIC versus models. There were several colors. How do we analyze that type of graph actually?
Okay, I will share my screen again.
Actually, I'm not at that field, but it's very interesting. That's right.
So here what I did was, so I had 63 models that I wanted to compare. And I have represented each model using one point, but the different circles. So this one is one model that contains all the parameters, the clinical, cognitive, psychiatric, and the volumetric. In this model, I have all the parameters. And in this model, I don't have the SDMT, but the rest of the parameters. So for an example, let's if I take these two models, so I have all the variables and I simulate the multivariate linear mixed model, and I get a DIC value for that. And for this model, I have the five parameters, and I simulate the model, I get one DIC value. So what I have plotted here is those DIC values for all the 63 models, so that I can compare the DIC values. Is that answer your question, sir?
Yeah. Okay, thank you. Right. Any more questions? Okay, thank you very much, Dr. Pubuditha. If no more question, I would like to invite formally you vote of thanks to Dr. Pubuditha, who is the Joint Secretary of IPSL. Dr. Pubuditha, over to you.
Thank you, Professor. On behalf of the IPSL Council members, and all other participants in this event, I would like to convey my sincere gratitude to Dr. Pubuditha. Dr. Pubuditha, your talk was really interesting and we learned about the disease and its progression. Thank you so much again, and I wish you all the very best. Later, we have to talk about the organizing committee of this event. Without their support, this event would not be a success. Thank you so much for your excellent support in organizing this event for us. At last, we would like to thank all the participants of this event. Thank you all again, and have a good evening and have a good weekend.
Thanks. Thank you very much.
Thank you very much for inviting me to give this presentation.
Thank you very much, Pubuditha.